Good morning, everybody, or good morning in the States, at least good afternoon here. It gives me great pleasure to introduce this session on the gut-brain axis, essentially, the role of prebiotics and probiotics in brain function. And that's an area I think that only a small number of years ago we would have thought was pretty fanciful, but now the evidence is growing. And I'm delighted to be able to introduce someone from my own location here at APC Microbiome Ireland in University College Cork, Dr. Harriët Schellekens. And Harriët, I was going to describe as a rising star in this area, but in fact, I think it would be more accurate to describe her as an existing star in this area. And so, Harriët, without any further ado, I'd like to pass over to you, please, and give your presentation. Thank you.
Well, thank you, Colin, for a lovely introduction and compliments. I am delighted to be here to be discussing some of my work and also others' work with the real experts. So I'm a lecturer in the University College Cork and, as Colin said, a funded investigator within APC Microbiome Ireland. And my work really focuses on the impact of nutrition and diet and the microbiome on human health, but particularly on metabolism and the regulation, the central regulation of appetite and food intake, and how this is changed under conditions of stress. So today I'll be talking to you about how we can target the microbiota-gut-brain axis for metabolic and mental health and stress-related conditions. And I will summarize some clinical evidence for both metabolic and mental health, and I'll take you to some approaches that myself and my colleagues have taken to lead to the identification of pre and probiotics for these two areas, using a screening, cellular and biochemical screening approach to select the best strains to go forward in animal and human. If you want to get in touch with me, here's my Twitter handle. And please, I look forward to discussions later on today, but also outside of this lovely symposium. We really are lucky in the era that we've been living in. For the last decade, and you can see here, this is what I looked at yesterday on PubMed, the field of microbiota have really undergone an explosion, and our knowledge of the human microbiota and microbiome have increased significantly. Now, for this audience, I do not need to go through this infographic. We all know that we have more microbes than we have stars in the galaxy, 150 times more microbial genes than our own genes. But what is really important in the field going forward, I think, is that each individual has their unique microbiota with their unique needs, as personal as a fingerprint. Also, in the times that we're living in now, it is perhaps a nice sentiment to realize that we never really walk alone. In the last 15 months under COVID, we've been under social isolation, but we've always had our microbes, and we need to take care of them. What we know now is that the microbiota is a critical mediator of gut-brain axis function, and there's lots of preclinical and clinical data out there. In my group, I really investigate the gut-brain axis in the context of metabolism and the central regulation, the short and long-term central regulation of appetite and food reward. But of course, the gut-brain axis is also really important for our stress response and mediates our mood and anxiety. And it really highlights the link between mood and food, because if you realize, or if you're stressed or anxious, your food intake is often also altered. And what was intriguing me when I set out almost a decade ago is that the gut hormones that govern our appetite, including ghrelin from the stomach and GLP-1 and PYY and CCK from the gut, and the neuropeptides, those that govern appetite, that they also innervate the hypothalamic pituitary axis, and that we have overlapping neurocircuitries that regulate food intake and our stress response. Also, obesity itself is an environmental risk factor for stress, anxiety, and depression. And we know now that the gut microbiota significantly contributes to our metabolism, and changes in microbiota composition and diversity have been linked to the development of obesity and stress. And remember the first study shown by Jeffrey Gordon that showed that human obese microbiota, independent of diet, is sufficient to confer an obese phenotype in mice. So the microbiota-gut-brain axis can be placed at the interface of metabolism and food intake and mood and stress conditions. And the mechanisms thereof I'm trying to investigate in my lab. So we know that the microbiota is a mediator of gut-brain axis function and that many factors influence this axis, diet being the most modifiable one. And this microbiota-gut-brain axis has an impact on different functional outputs, from food intake to cognitive behavior to stress, social interaction, and fear. And this is the basis of the therapeutic targeting of the microbiota. Of course, throughout this symposium, we'll see lots of evidence of using probiotics, symbiotics, postbiotics, prebiotics for the therapeutic targeting of the gut-brain axis functioning. But we should add now to this list also psychobiotics, which is a term, a novel class of psychotropics, a term coined by Professor Dinan, Stanton, and Cryan, and defined as live organisms that, when ingested in adequate amounts, produce health benefits in patients suffering psychiatric illness. So these novel types of probiotics, psychobiotics, will also likely be able to be used in stress-related conditions such as anxiety, and they started somewhat of a psychobiotic revolution. But what is clear is that the microbiota represents a targetable therapeutic option in metabolic and in mental health. And I've tried to summarize some of the clinical studies that support the anti-obese effect of particular microbiota strains, so probiotics as such. I won't quote through all of them, and there's many more. A lot of them, I must say, also with negative results. But some of the positive ones here is this study in 2014 using the probiotic VSL3, which was a multi-species probiotic, about 7 strains, that showed in using 60 individuals in 4 groups that this probiotic mixture confirmed some anti-obese benefits, including insulin, improving insulin response and improving inflammatory profile. Then a more recent one here on the right. This is a proof of concept exploratory study in the group of Patrice Cani et al. That used Akkermansia and showed that there were some benefits as well, particularly improving insulin sensitivity and some improvements in weight gain, but which were not statistically significant yet. And then here, the one in the bottom, which was in the bottom right, which was published in 2020. This again is also a multi-strain showing that some improvements in postprandial glucose in subjects with type 2 diabetes. And this is the clinical evidence that led to pendulum probiotic, which is now used for diabetes. But we don't have a lot of the mechanisms behind how a microbiota actually confer these anti-obesity effects. Now, what are these microbiota-driven mechanisms of food intake and appetite? In 2017, we wrote this review, which summarizes some of the mechanisms by which microbes can confer changes on our appetite and food intake. We can think, for instance, our microbes will ferment our foodstuffs, and especially indigestible fibers, to short-chain fatty acids, which have lots of beneficial effects, but also neuroactives such as GABA and serotonin. Other bioactives and metabolites may activate receptors which we have in the gut, for instance, receptors on the anterior endocrine cell lines, which can lead to changes in gut peptide secretion. Metabolites can also modify the immune response, and perhaps a lot of undiscovered bacterial neuroactives are suggested to be able to reach the brain and have central effects via either vagal nerve modulation or reaching the brain via the circulation, and in that way modulate appetite and satiety centers in the hypothalamus. But what is clear from these mechanisms is that if you do not have the right microbes, this can set the stage for the development of obesity and diabetes and anxiety throughout life. Now, how can we use these mechanisms and what we know about how microbes do their thing as such to identify microbes as probiotics? me and my colleagues, we've set up a pharmaceutical approach to mine these microbes and metabolites to bring these bugs to drugs. As you can say, by creating a pipeline or a platform of several cellular and biochemical assays to interrogate the APC's culture to product platform, which is a continuously expanding, well-characterized biobank of microbes for their probiotic, functional, and psychobiotic effects. We can look, for instance, and screen for immune response, intestinal permeability, their ability to secrete short-chain fatty acids, their ability to modify tryptophan metabolism, and we also look at the metagenomic data for their putative ability to modify brain function. And then we can identify the most promising bacterial strain for further in vivo and ex vivo studies and ultimately clinical trials. Because you need to have a system that can select the best and most promising bacteria first. So using this targeted approach, we can identify functional bacterial strains while at the same time gaining mechanistic insight into the gut bacteria-derived metabolites as modulators of gut-brain axis function. Now, in my group, I'm particularly interested in the ability of bacteria and their metabolites to modify G-protein coupled receptor signaling across the gut-brain axis. G-protein coupled receptors are C7 transmembrane spanning receptors that couple to a G protein and then have further downstream signaling. We have about 5 to 6 classes of GPCRs, totaling up to about 800 different G-protein coupled receptors, some of which are orphan receptors. And in the last few years, from 2017 onwards, only a handful of studies have highlighted the potential mechanistic link of microbes and their ability of their metabolites to modify this G-protein coupled receptor signaling. And if you realize that currently 34% of the FDA-approved drug are actually targeting about 108 of the total of 800 GPCRs, it just shows what an untapped, unexplored reservoir bacteria metabolites present for the modulation of this GPCRO and modification of gut-brain axis function via modulation of these GPCRs. Now, a particular G-protein coupled receptor of interest that we've been working on is the Ghrelin receptor. So this is one of these G-protein coupled receptors, the Ghrelin receptor, which is activated by its endogenous ligand Ghrelin that is produced by the stomach. And this has multiple functionalities, both in the periphery, which include gut motility, glucose homeostasis, and immune modulation, but also centrally, the energy homeostasis and appetite regulation, hedonic food intake, and stress response and effects on mood and anxiety. So Ghrelin signaling in the context of obesity is really at the interface again of mood or food intake and metabolism and mood. And these receptors are expressed in the stomach and in the gut, as well as in appetite centers in the brain, in the hypothalamus, reward centers in the brain, such as the ventral tegmental area, nucleus accumbens, and also the hippocampus. And it has been shown that peripheral Ghrelin can reach the brain at the level of the median eminence. And what is interesting in obese individuals is that this cycling of endogenous levels of Ghrelin, which spike before each meal, which initiate food intake, that this spiking in obese individuals is absent. And most notably, the increase in nocturnal Ghrelin is absent in obese individuals. Now, we started to look in literature, is there a role for microbiota in Ghrelin signaling? And we published this in February of this year, which highlights some evidence from the literature on how gut microbes and their diet-derived metabolites and native metabolites can modify Ghrelin secretion in the gut, and in this way modulate Ghrelin's functionality from energy homeostasis to adiposity, as well as stress and reward signaling and cognition. And one of these findings that we showed was that in germ-free animals, which are the animals that grow up without any microbiota present, which are characterized by heightened stress response and altered anxiety-like behavior, but at the same time are hyperphagic while being diet-induced obese resistance. So they're really messed up. And these germ-free animals actually also have a decreased circulating levels of ghrelin. So we asked ourselves the question, can we target the ghrelin receptor, this GPCR, via the gut microbiome? So we used APC's culture collection and screened cells that were expressing this ghrelin receptor to see if the metabolites were able to modify ghrelin receptor signaling. And this was published in ISAPP in October in 2019. I won't go into the specifics of it, but we were able to show that Bifidobacterium longum were able to attenuate ghrelin mediated signaling through this receptor. We could look at the trafficking. We were able to show that specifically the Bifidobacterium that were high acetate producers were able to attenuate ghrelin receptor mediated signaling, and confirmed that acetate itself also had this effect. And we also confirmed that this Bifidobacterium longum APC 1472 was able to attenuate ghrelin mediated beta arrestin signaling and also downstream phosphorylation of ERK. So we chose the most promising one, Bifidobacterium APC 1472, and we wanted to see and validate if this BIF was able to confer anti-obese effect in an animal model of obesity, mice that were fed high-fat diet, as well as in a cohort of overweight and obese humans. And I'm going to the punchline. This was published this year in January 2021. And as you can see, there's a lot of authors involved. It was a real team effort, so credit to everyone involved. But to summarize the data, we showed in the high-fat diet fed animals that 16-week supplementation with the BIF longum in their drinking water was able to attenuate weight gain mediated by the high-fat diet, and specifically reduced here in the bottom left, specifically reduced individual fat depots and reduced leptin. We also showed that there was an improvement in glucose tolerance in the high-fat diet fed animals with this Bifidobacterium longum supplementation. And surprisingly, we also saw that there was a reduction in the stress hormone corticosterone. So again, perhaps linking this mood and food. So next we wanted to see, taking this strain now from screening to animals and then into humans, what would happen in a cohort of overweight and obese humans. So we performed a randomized double-blinded parallel placebo-controlled study on 124 individuals, so very well powered, and they were all overweight and obese and had a BMI of between 28 and 35. And they're receiving 10 billion probiotics in capsule format per day. Now, the primary outcome was body mass index. We did not see any changes here. We also didn't see any changes on the waist to hip ratio, but we did see a significant reduction in fasting blood glucose. And to highlight that all the overweight and obese individuals within the study had normal fasting blood sugars of below 5.6 millimole per liter. So a reduction of 0.27 would make the difference between becoming pre-diabetic or even diabetic. And none of these individuals were pre-diabetic. And this reduction in fasting blood glucose corresponded with an increase in the Bifidobacterium abundance following the administration of the BIF longum APC 1472. What we did next is a subpopulation analysis. So we looked at only the obese individuals, and we saw that the fasting blood glucose effect was even further or more pronounced. We also saw in this obese cohort that there was a reduction in cortisol awakening response, which is the main readout of the HPA axis functioning in humans. And we saw that there was an increase in fasting active ghrelin levels. So to summarize this study, we see a dual effect of reduction in blood glucose and a reduction in cortisol, which parallels what we saw in the high-fat diet fat animals. And we see an increase in ghrelin. Now, this increase in fasting ghrelin might be paradoxical because doesn't ghrelin increase hunger? But if you remember this graph in obese individuals, notably the nocturnal rise in ghrelin is absent. So the rise in fasting ghrelin levels here are likely to indicate a normalization of ghrelin dynamics in these individuals. But what these findings really reinforce is the concept of the link between metabolic disease, mental health, and the potential to ameliorate both via microbiome-targeted approaches. Now, for the second half of my talk, I would like to take you through some other findings, and in this instance with prebiotics. So in this study, also a model of obesity, diet-induced obesity in the mouse, we used fructo and galacto oligosaccharide supplementation. And we did not see any effect on body weight gain. We didn't see an attenuation of body weight gain, but we did see a significant reduction in the individual fat depots and a reduction in leptin. And when we looked at the anxiety-like behaviors using an elevated plus maze, which you can see here on the bottom right, it's an elevated plus maze with 2 closed arms and 1 open arm. And this is an anxiety behavioral paradigm that measures the anxiety levels of the animal. And the more time the animal spends on the open arms, the less anxious it is. And we can see in the high-fat diet fat animals, the red bar, supplemented with FOS and GOS, that there's an increase of time spent in the open arms, indicative of an anxiolytic effect. In another paradigm of anxiety, using the open field, we measured the latency to go to the center. And we can also see that the FOS and GOS supplementation decreases the latency to go to the center. And we saw a reduction in corticosterone, but only in the low-fat diet animals. Now, my colleagues have used this FOS and GOS also in a chronic social unpredictable stress model. And they showed here that the FOS and GOS supplementation was able to restore deficits in social behavior. If you compare here in figure B, the gray bar to the red bar, there's an increase in social interaction. And also on the cognitive aspect, there's an improvement in novel object recognition. And then the lower figures, again, indicate also improvements on anxiety and anxiolytic effects, because supplementation of FOS and GOS in the stress group, again, increases the time spent in the open arms. The FOS and GOS supplementation in the stress animals was also able to significantly reduce the corticosterone, the stress hormone, and an increase in Bifidobacterium again was observed. What is also interesting in this study is that the FOS and GOS supplementation was able to reduce the interleukin-6 response and TNF-alpha response to CON-A. So it's demonstrating a positive effect on immune profile in response to stress. And similarly, looking at immune modulatory effect of prebiotics, this is a study that I was involved in, which uses an oligofructose enriched inulin. And this was shown here in the top right graph to ameliorate and reverse stress-induced immune priming in the periphery, so in middle-aged rodents. So you can see here that middle-aged rodents have an increase in percentage of neutrophils, which is attenuated when they are supplemented with this inulin. And on the bottom figures, we also showed that supplementation with this oligofructose inulin was able to attenuate age-related microglia activation in the whole brain. So this perhaps shows that fiber, inulin, you find that in leeks and is a fiber, that fiber, not fast cars, are the answers to your midlife microbiome crisis. But of course, this is also relevant for obesity, because in obesity, we have a chronic low-grade inflammation and also hypothalamic and neuroinflammation. Now, what is the clinical evidence for potential using prebiotics to alleviate stress? This study here on the left in 2015 by Burnett et al. Also showed that FOS and GOS, or in this instance GOS only, was able to decrease a cortisol awakening response in healthy volunteers. And in 2020, a more recent study, short-chain fatty acids, which are a byproduct of a lot of these prebiotics, it was shown that the colon delivery of short-chain fatty acids was able to attenuate cortisol response in psychosocial stress in healthy men as well. Now, this is a really interesting study in 2021 by Nathalie Delzen that used a randomized single-blinded multicentric placebo-controlled trial in 106 obese individuals, and they received again an inulin, in this case extracted from chicory root, and they were able to show that this inulin supplementation in obese subjects had moderate beneficial effects on emotional competence and cognitive flexibility, but that this was subject to positive responders and negative responders. And when they looked at the microbiome of the positive versus the negative responders, and these were all obese, they showed that the microbiota profiles were very different. And specifically, they showed a specific microbial signature in the positive responders, which included here in figure C, an elevated coprococcus levels at baseline. And that if this was present, that these individuals were more prone to benefit from the inulin prebiotic supplementation in terms of mood. They also demonstrated, following the supplementation with the inulin, that there were increases in Bifidobacterium, but only in the positive responders, and that this also correlated with improvements in IL-8, reductions in subcutaneous fat, and reductions in dipeptidyl peptidase 4, which degrades incretins such as GLP-1. So these levels of could perhaps predict or mediate the beneficial effects of inulin on behavior in obese individuals. But overall, what this shows is that inulin intake may improve mood in obese subjects exhibiting a specific microbial fingerprint. So this is something I think the field is moving towards as well. Now then, what is clear is that diet composition and nutritional status are the most critical modifiable factors regulating gut microbiota. We wrote these two reviews, again highlighting the mechanisms, but in this case, the interaction of diet with microbiota, inferring on the mechanisms of microbiota on gut-brain axis function. So indicating that nutritional interventions targeting the microbiota through these specific mechanisms are key to maintain metabolic and mental health. Now, this is my second last slide, just another few studies on clinical evidence, and in this case, not prebiotics, so not diet, but actual psychobiotics. So evidence in humans of bacterial strains with positive effects on mental health. This study here showed the beneficial effects on depression, symptomatology, and metabolic status following 8-week multi-species probiotic in 40 patients with major depressive disorder. And this study in 2010 also, which was a double-blind placebo-controlled randomized parallel study, and this showed alleviated psychological distress in 55 volunteers following 30-day supplementation with these two probiotic formulations. This was a randomized control trial using a multi-species, again, 7 strains, but all of these studies all include BIFs. This particular one included a BIF and a BIF lact, and 7 strains in total, but only in 20 healthy individuals, but reduced overall cognitive reactivity to sad mood. And then a study from APC itself, using a single species, again, a Bifidobacterium longum, which was able to reduce stress and improve memory overall. And a study by another group that continued on this, demonstrating modulated neural responses during social stress. and brain coping centers were being activated. But it doesn't always work. This is a study from our group, which initially showed positive effects in animal models, but did not translate into human population. But taking into account that this is a study that was done in healthy male subjects, and you can't fix something that isn't broken. So just to end with perhaps some challenges of the current field, that both looking at studies currently of metabolic and stress that there is not that much data yet. Most studies are small, observational, and usually do not have a dose. They use as much probiotic bacteria that they can. There's no consistent readouts. The mechanisms are not always completely understood, and microbiome and metabolites are rarely measured. So we really need more integrated nutritional interventions, and this was a manuscript that we wrote with our collaborators, highlighting some where the field should go in human studies to study the effect of nutrition or probiotics on the usefulness in affective disorders. But what is clear is that we need more mechanistic studies and human interventions. And then I will always come back to one of the quotes by Hippocrates that I used in my thesis defense 9 years ago. Let food be thy medicine. And moving forward, we know that let food be for your microbes, be your medicine. So, of course, I can't finish without thanking my current and present lab members, and for the e-biomedicine study in the Lancet, particularly Christina Torres and Marcel van de Waal, and of course, my collaborators. So, thank you very much. Colin, you're mute.
Thank you so much. That was a wonderful talk, and there are so many questions that I had noted down myself, but I think I'll come and find you myself afterwards to ask those ones. There's I think a healthy, and I think you'd agree with it, a healthy kind of skepticism about this area because it's so complex. I mean, there's so many readouts when you look at these trials, either in preclinical models or in humans. What's your reaction to that, I guess? You know, is the evidence really robust, do you think? Or are these still hints at mechanisms and at activities and functionalities?
Well, I think that's a two-part question. Is the effect of microbes on our metabolism and mental health real? Yes, I think so. I think specifically in clinical studies, there's so many variables and mediators and confounders that if a study might work one time and not the other time, it can depend on these confounding factors. In relation to mechanisms, I think it is not, it goes back to the saying, it doesn't matter who is there, but what they actually do. And I think there are multiple bacteria that can do the same thing depending on their circumstances again. So depending on what type of diet, if you use a probiotic and you do not have the right diet to make the correct metabolite, then you might not see the beneficial effect. And also in this study that I showed, where they really showed that there were responders and non-responders, depending on the baseline microbial signature. I think that is something we need to start taking into account with all the studies that we're going to do right now.
And you touched on it a little bit in your answer there, but for every microbe psychobiotic, which is a term I hate, by the way, but anyway, and for every positive result, are there like 10 strains that give no result or negative results? Or, you know, what's the frequency with which things work in these preclinical models in particular?
Well, I don't think we can answer that just yet, because there's not enough large clinical trials done. Most studies are small and have only a few subjects within each group. So I don't think we can answer that just yet. But if you look on clinical trials and randomized clinical trials as well, there's just as many with different strains that are negative than the ones that are positive.
That's kind of encouraging. I don't like seeing any situation where everything works.
Yes.
Bob Hodkins has a question there. In the Belongum human study, were there changes in microbiome composition or diversity recorded in that study?
That's a good question. Unfortunately, due to time, I couldn't show everything, and I just wanted to make a more conceptual story, but there were no overall changes in microbiota composition, not in the animals and not in the humans, but we did see an increase in relative abundance of BIF.
But that would be in the fecal microbiome, I guess. I mean, there could be changes farther up the GI tract that we wouldn't be seeing.
Yeah, for rodents, it's cecum, and for the humans, it's fecal.
And the last question, because we're running out of time, but where do you think in the GI tract the greatest communication with the brain is? Is that, you know, we hear a lot about the vagus nerve, but is it the upper GI tract, the lower, you know, where do you think the most of this communication is happening?
I think there's different types of communication across the digestive tract. I think in upper, it's perhaps more vagal. I'd just be guessing now, but of course, the different mechanisms would both be activation of vagal afferents as well as metabolites that can have local effects or perhaps via indirect effects reach the brain. So I think that's, I don't think it is where is it most active. I think it is based on the different mechanisms. And I also think that individual strains may well have different mechanisms at the same time that contribute to a health effect and improvement of gut-brain axis function.
Yeah, that makes sense. And so, Harriët, once again, thank you so much for your presentation. I really enjoyed it. And I'll pass over now to Eamon Quigley to introduce our next speaker. Thank you.